and welcome to SciLux, the podcast where we talk about scientific developments and technological changes in Luxembourg, but our listeners are already used that it's not always in Luxembourg. Sometimes we have episodes where we have international guests alone, and sometimes we have episodes when we have international guests who cooperate with researchers based in Luxembourg. This is the case today, and it also means that we are proudly powered by Research Luxembourg and recording at the Media Center of the University of Luxembourg. And now on to the guests. So we have two guests today, and the first one, sitting just next to me, is Carole Linster. And Carole is an associate professor in biochemistry at the Luxembourg Center for Systems Biomedicine, for those of you who know LCSB, at the University of Luxembourg. She leads the enzymology and metabolism group, and her focus is on rare metabolic diseases. And then we have joining us from Seville, Rafael Fernandez Chacon, who is the director of the Institute of Biomedicine of Seville and professor at the Department of Medical Physiology and Biophysics, as well as a cybernet investigator. He is interested to understand the way synapses work in the context of health and disease. Carole and Rafael, thank you so much for coming today.
Thank you, Anna, for the invitation.
Thank you, Anna. Great to be here.
Pity that you're not personally here. We should have recorded it because I think you were in Luxembourg a couple of months ago, right?
Oh, yes. It was a great experience. I'm looking forward to come back soon.
I would prefer to go to Seville, actually. I've never been, and this is absolutely on my bucket list.
You're more than welcome to come. We are so happy to have this relationship between Luxembourg and Seville. We are really looking forward to keep it alive for many years, okay? So it's going to be great.
That's very good. So maybe let's start with the relationship, actually. How did it start? How did you say, okay, now LCSB needs to cooperate with Seville? Carole.
So I think it was not long after we had our new director starting, Professor Michel Hennecker. He had already cooperations with the IBIS in Seville, and so he organized a meeting between the group leaders of the LCSB and the group leaders and the director of the IBIS. And so all the PIs were briefly explaining a bit what they were doing. Of course, our management had already identified complementarities in our expertise in the methodologies that we are using. And while we were telling our stories, our focuses in research, we, especially Rafael and myself, we identified very nice complementarities and that we have very common interests in certain diseases. So we started this project.
How do you define complementarities, Rafael? Does it mean that you have labs that can do something in Seville that we can't do in Luxembourg?
Exactly. I mean, just to continue with Carole's description, when we were discussing at the meeting, we realized that we have common scientific questions. Common scientific question based on a general group of diseases that are lipofuscinosis. So Carole had done a great, very interesting work in lipofuscinosis using zebrafish model, a little fish. She's an expert on that model. And we were and we are interested in this kind of diseases, but in mouse models. So then immediately we realized that we have common interests and we could even focus common mechanisms to advance in the knowledge and the future therapeutic approach to treat these diseases. And on top of that, we found that indeed we have, from the methodological point of view, important complementarities. In our lab, we know a lot about mouse genetics, how to analyze them using, for example, electrophysiology, that these are techniques that allow us to look how the neurons behave, how the neurons communicate with each other. We also use microscopy. We use electron microscopy. We use proteomics to understand what is the repertoire of proteins that change in a disease. And Carole has another complementarities.
Yes, exactly. So Rafael talked about mouse and we are using zebrafish. So these are complementary models. They have each their advantages and disadvantages, but they have the same genes for many of the genes. So we can study the same diseases in those models. And also, Rafael talked about proteomics, studying the whole complement of proteins in a cell. We are looking at smaller molecules, the metabolites that are the intermediates of our metabolism. So the process where we degrade our nutrients, build up the compounds that form our cells. So we have the methodology and the instruments to look at those small molecules, and again in a comprehensive way. So that's why we're talking about metabolomics. And this is very complementary because the proteins form and act on these small molecules, and we are looking at the small molecules. So looking at both gives us a more comprehensive picture of for example, the status of the disease, what the disease is changing in our models.
We got very much deep in already, I already have a lot of terms that I need defined, and I want to, of course, discuss the mice and the zebrafish, all this. But before we do that, I always wonder how it starts, right? So let's say you just want to start with a gene, or how do you discover this? How do you confirm, okay, this gene is responsible for that? Rafael, how does it work?
Thank you for the question. This is interesting. I'm going to tell you about two perspectives. So in general, when you discover that a gene is causing a disease, this normally starts at the clinics. And this is one example, one of the diseases that we're going to talk about today. There is this disease called Kuff's disease. Indeed, this disease was described 100 years ago, indeed in 1925. And it was a disease that they discovered that is a disease that the patient, young patient, around 30 years old, they have a psychiatric problem, they have dementia, they have movement disorders, and later they have early lethality. And they already detected how the disease was inherited. And then that's happened, 100 years ago. But later on, until 2011, in this case, this disease was a disease without knowing a knowing cause. So it was several groups internationally. This Kuff's disease, this is neuronal ceroid lipofuscinosis, autosomal dominant, that from the adult. Three groups discovered: the group from Moch in Prague, the group of Cruchaga in St. Louis, and the group of Cosette in Montreal. They use DNA, genomic DNA from the families of these patients, and they read the sequence of the genome. And they compare the genome of the patients with the genome of the other brother and sister that they didn't have the disease. And they discovered the difference between these two genomes. The genome is the collection of genes. And they discovered that one particular gene had some particular mutations. The gene I'm talking about is DNA JC5. That is a gene that encodes a synaptic vesicle protein. That is a protein that we are working with. So that was the point in which I tell you the example that how do you discover the gene? So you have the disease, and later on you discover it. But what happened when you discover the gene? When you discover the gene, you want to know why, introducing that mutation in the gene, you have the disease. And I must say that this is when we came into place, because I started to work with this cysteine protein, that this little protein, long time ago, when I was a postdoc in Tom Sudov's lab in Dallas in the United States. And at that point, we didn't know that gene could be connected to a disease. But we were trying to understand the genes or the proteins that are in one synaptic vesicle, in one nerve terminal. So after many years, this research is going together to a very important common point. Knew a lot about what the protein is doing in a normal nerve terminal, and now we are trying to understand what is happening when the protein is mutated and causing this disease.
And I know that for you, Carole, as well in the lab, right? Sometimes you study a gene before you actually know that it's related to a disease.
Yes, exactly. And I like that a lot in Rafael's description, because it emphasizes how important it is. And I always try to take the opportunity of these kind of podcasts to say how important fundamental research is, because, we are studying these genes, and when we know what they are doing, and then now with the genome sequencing techniques, we can very rapidly find out, which gene is mutated in the disease. And I mean, sometimes it's more complicated, but it's becoming much more straightforward to find out about that. However, if you find that and then you don't know anything about the gene, you are not very much advanced, In our case as well, there is a different disease where we had studied the gene for several years, and we had really figured out what it's doing in metabolism and everything. And then children came into the clinic with all the normal suspect genes being completely normal, but this gene being mutated. And, if it had been found 10 years before, nobody would have had a clue what this could do. And because we had in the meantime understood what the functioning metabolism is of that gene, we could very rapidly work with the clinicians to confirm, first of all, that this is indeed the gene whose mutation leads to the disease. And then actually in that case, what becomes deficient is a certain vitamin. And by giving this vitamin to the children, they were doing much better. So not only did we understand, but we could really tackle the disease by the knowledge, the fundamental knowledge we had accumulated before.
Exactly. This is this very, very important point that I love emphasizing during the podcast, especially and not only any kind of discovery, in fundamental science we don't really know what it's going to be used for, but it's useful because later on there'll be someone who comes up with a solution, right?
I would like them to emphasize that concept. I mean, I completely agree with Carole how important it is to emphasize it. And I want just to add a little piece of information to the story of the gene that we are working with, because, the fundamental question that we had a long time ago is, which is the function of this gene? We knew that this gene were important for the nerve terminal function. And in order to do that, we generated, as I told you, more than 20 years ago, we got the genes and we produced mice without this gene to understand the function. And one thing that we discovered is when we removed this particular gene, now that we know that causes the disease, we discovered that the nerve terminals degenerate. And we didn't know why. Even some people told us, why are you studying this gene? Why don't you go directly to a gene that that is linked to a disease? I said, well, look, at this moment, we don't know if the gene is going to be linked to a disease, but we know that it's going to be important, because when you remove it, the neuron and the synapse is failing. Is coming down. So we need to understand that mechanism, because it's going to be important. There is one important question is, all our neuronal connections, they are for many, many decades here. That's the reason, because we can remember, and we don't know how those nerve terminals are maintained. But what we realize is when we remove it, the nerve terminal is not maintained anymore. So that's a highly relevant question I wanted to understand. So later on, years later, it came the discovery of this disease. So then when we started to work, we had already an important advantage to advancing the knowledge of this.
And now I think it's, as Carole has already mentioned a little bit of what she specializes in, and the same for you, Rafael, I think it's the moment to set a little bit the context for, the numbering, let's say, part. So the time to ask the pub quiz questions. So from Carole is about the genes, and from Rafael, it's more about the synapses. So if you can both ask the questions, and listeners, remember, of course, the answer only at the end. And I always say, don't Google, continue listening to us. We will reveal the answer later on. So Carole, if you can start with your question.
I'm going to ask a question about, indeed, the answers will be two numbers. And I'm glad we talked about genomes already, so we have a little bit this concept. So since 2003, we have cracked the code of our genome. So we know the entire sequence of the human genome. And actually, since then, we also know how many genes we have in this genome. And actually, each gene encodes for a protein. So by that, we also know how many different proteins we have. And so my question is how many genes do you think we have in this human genome? And for how many of these genes do we still not know the function?
Very interesting discussion, and I'm sure a very interesting answer too. And for you, Rafael, we said synapses, right?
Good. So, we're talking about the brain. So the brain capabilities rely on neurons and how neurons communicate with each other through synapses. So the question is related to the number of neurons that we have in our brain and the number of synaptic contacts that we have in the human brains. So that's my question. How many neurons do we have roughly in a human brain, and how many synaptic contacts are formed by those neurons in the human brain?
And actually, it made me wonder already, probably a stupid question, but I need to understand, what's the relationship between the genome and the brain? Because I thought we were talking about the genome, but now we are talking about the brain.
Okay, Carole.
Yes. So as I said, like the genome contains the gene, and each gene encodes for a protein, and those proteins are basically carrying out all the functions that we do in our organs, including the brain. So a number of these genes are extremely critical for the functioning of the brain, and that's the connection. And as we talked about genetic diseases that specifically affect the brain, so those genes in particular, we know that they are very important for the functioning of the brain.
Now I get it. I was afraid that I will not understand the answer, but it was absolutely clear. I sometimes complain here a bit about the educational system. I know I should not, but still, it is just so unrelated the way we study biology, right? You do the brain, and then 3 years later you have genetics, but you just really fail to see the connections. That's why, I couldn't really perceive them myself either, right? Okay, but let's not complain too much about the education system, I think, because, we are not specialists here and we all have our opinions for sure. Okay, so you mentioned already the mice models and the zebrafish models. Let's start actually with mice, because I think people know more about them, but I guess every single lab has their own specificities and specific models. So what is your model about, Rafael?
Yeah, so our model indeed is there is the mouse, because, the mouse is a mammal, and there's many basic mechanisms that are very similar to what happens in humans, although it's true that it's not the same a mouse and a human. And also we can use the mouse not only to investigate the mechanism, thanks to genetic modification of the mice, and we can introduce the mutation in a patient, we can introduce it in a mouse. In addition, we can use the mice for preclinical research. So once you have discovered the mechanism and you want to test a particular drug or particular therapeutic approach, then you can do the preclinic in the mice. This is very important. I think that now we're talking about experiments with animals, because we need to explain to the audience how important this is. So, there is some people that they have some doubts if we should or not do experiment with animals. And I must say that the answer is yes, and this is very important. And some people are scared. They think that the mice can be suffering. And I need to convey the message that this is really, really under control. Every mouse every experiment is under ethical control, and it's for sure that the mice, they are not suffering. This is really important. Every experiment is done. The well-being of the animals is a high priority, and this important concept. I can tell you that there is a European initiative. They're called the transparency. Every year, the labs and the institutions, they open the numbers of the experiments that we are doing and try to convey to the public how well done this is. So they need to make sure that what we're doing is really responsible. And in any occasion that we can use a model that is not a mouse model, for example, is cell cultures, we try to do it. But it's true that in the case of the brain, we really need this mouse model, because the brain is a very complex organ, and we can study the brain properties only using living models.
It's great you also mentioned that, because every type of science has its own characteristics when you're going out to the general public. I specialize a little bit in space science, and in space, of course, you're discussing bringing back all the amazing resources that are there, mining asteroids and whatever. I mean, that the general public is talking about, like helium-3 or rare earths or whatever. And it's always every single journalist will discuss that with the space scientist. And then when it comes to any kind of research, genetics and whatever, and especially mouse models, there's, of course, the discussion of ethics, but we have to keep repeating it because, people want to know that. And unfortunately, somehow it's still not in the public conscience that in Europe we really have a lot of controls, as you said, a lot of tests, and as you open the labs, and it's not like it used to be in the past. Yeah, zebrafish. To be honest, this is the first time I hear that there's such a model.
Okay. Well, however, we use it quite a lot since a number of years, and it has its particular advantages. Of course, it's a lower organism, let's say, than a mouse. However, it's also a vertebrate. It has a central nervous system. And the particular advantage, one of them is that the larvae of the zebrafish are transparent. So until a certain age, we can look in the microscope onto these larvae and see through them like all the organs that are developing. In addition, the organs are developing extremely fast. By 2 days after the fertilization of the egg, all of the organs are developed. So we can look at the development of these organs in real time, basically, under the microscope. In addition, the genes are, as I said before already, conserved. More than 80% of the genes that are causing diseases in humans are also present in the zebrafish. So we can model many, many human diseases in the zebrafish. We can, as well as for the mouse, we can also genetically manipulate relatively easily. Actually, in the case of the zebrafish, the genome, we can knock out genes, knock down. That means just eliminate a part of the function. And we can also put in reporter genes. So, for example, we can specifically fluorescently label certain neurons in the brain, dopaminergic neurons, for example, that are important in Parkinson's disease. And then during the development or after a certain genetic modification, see like what is happening with this type of neurons exactly in the brain. So for imaging, it's really a big advantage.
But then it also means that you're using both of your models to check something similar. So does it mean that you now are really getting rid of certain genes, the same genes for the same models? What are the genes and why?
Well, in particular, is this gene I mentioned earlier is called DNA JC5 that encodes a protein that is called cysteine string protein. So this protein is a protein that is in the nerve terminals, and the nerve terminals, we have little compartments that are loaded of small molecules called neurotransmitters. So let me tell you that this is called the synaptic vesicle, this little compartment. So when an electrical impulse arrives to the nerve terminal, the synaptic vesicle release these chemicals, these neurotransmitters, and they go to the neighbor neuron and then they transmit the signal. That's what's happening in the synapse, okay? So then this protein I'm telling you about that is in the vesicle, this cysteine string protein, is important to maintain this release of neurotransmitter. So when we remove it, then the release of neurotransmitting fails because some of these other proteins involved in the mechanisms, they are not assisted, So let me tell you that this cysteine string protein, the CSP, is what we call a chaperone. When you have a machinery that is functioning during a long time, you need to fix it. In some occasions, the proteins become a little bit sick, so you need a chaperone. It's like a doctor that goes there and somehow repair the protein, okay? And this is the role that we have found so far, and we are going deeper to understand what's going on. And of course, in the perspective of these diseases, lipofuscinosis, that Carole is investigating, we discover that, and it was discovered by others, that when you mutate this gene, you trigger this lipofuscinosis, because this is indicating that CSP is doing other things in other organelles that are called lysosomes, and then the Carole can tell you about that. So we share the mechanism that we are investigating in the lysosomes. We think that are going to be common mechanisms for different genes, the gene that we are studying and the genes that Carole is studying that she can tell you about.
Yes, exactly. So the disease that we are, a form of the disease Rafael is studying with the Kuff's disease, but there is a global term, or he already said the complicated one, neuronal ceroid lipofuscinosis, that refers to this. Actually, it's pigments that accumulate in our neurons that are mainly formed of lipids and proteins, and they have this fluorescence, so one can see them also by microscopy. I'm not sure, actually, the Kuff's disease is also called Batten disease, but many of these neuronal ceroid lipofuscinosis are called Batten disease. It's a more global and common term, let's say. And there have been more than 10 genes, I think by now 13 genes that we call the CLN genes, that all lead to a form of this Batten disease, let's say. And one of the commonalities is this accumulation of lipid protein pigments in the brain. And also many of these genes encode for proteins that are somehow indeed then associated to this organelle that Rafael mentioned, which is the lysosome. Lysosomes are smaller vesicles within the cell that are again delimited by a membrane. And within these lysosomes or vesicles, we degrade damaged molecules that are in our cells. So there are chaperones, as Rafael said, to try to fix them. But when you cannot fix them anymore, our proteins or lipids or also saccharides, they go into the lysosome and they get degraded there so that you do not accumulate damaged molecules. And so if something in that lysosome does not work, you start accumulating certain damaged proteins or certain damaged lipids or certain damaged saccharides. And that then leads to problems, many of them in the case of Batten disease, specifically in the brain. Other times, other diseases are caused by dysfunctional lysosomes that can go more problematic in the muscles or the heart. But for Batten disease in particular, it must be molecules, damaged molecules that when they accumulate, they specifically damage the brain.
But when we talk about diseases that are caused, when you confirmed by one gene, is it always going to be one gene?
So no, it's especially in the ones that we are talking about here, these diseases that run in families and rare diseases, indeed, they are monogenic. So they are caused by a mutation in one particular gene, a mutation that often really modifies the function of the gene so much that then it leads to an acute form of a disease. But other more chronic and more common diseases are often caused by less harmful mutations in several genes. And the fact that you have, unfortunately, combination of several mutations in those different genes then can also, in a longer time frame, lead to diseases that come with aging. But the ones that we are talking about here, these Batten disease forms, they are caused indeed by mutations in one particular gene. And that's why you can also do this linkage analysis in families where you see, okay, this brother has it, the sister doesn't have it. So let's look now where this particular gene is that is different.
For me particularly, it's difficult with the naming to remember all those, long names and whatever. And, obviously names of diseases, as they are often named after someone who discovered them first and whatever, that's a bit easier because it's still a name of a person. But I particularly like the word chaperone to be used in this context. And I just wanted to ask you, Rafael, about the chaperone. Is it something that we can actually create artificially? So can we have chaperones, so like some kind of medicine that we could introduce then?
Some people are trying to develop chemical chaperones, and this is true. There is two major questions about the chaperones. First, we need to understand that the chaperone is a protein that is going to help other proteins. So the question, and those proteins are client proteins, so those questions are client proteins. So when you have a problem with the chaperone, one question that you have is, which are the client proteins? Because when you remove the chaperone, when you remove the doctor, you need to see which are the clients. So that's an important question. So when do you know the clients? Then so you can design specific targets to do something to replace the client and so on. And the second thing is to understand what is doing to the client. What kind of damage is producing. Is probably normally the problem is that those clients become unstable. They become unstable and then they disappear. Either they can form aggregates or they can be degraded. The cell has specific system to degrade the proteins that are not functioning well. What is a good thing, because otherwise they will be interfering or forming aggregates. So what I'm trying to convey you that this chaperone concept is very important to understand because it opens a number of different possibilities to do therapeutic intervention. Yeah.
Okay, as I said, it was naming time. So one more question about naming of the genes. The CLN genes we were talking about, why are they CLN?
Actually, I'm wondering as well, because I think it's like an inversion of this NCL. So NCL stands for the neuronal ceroid lipofuscinosis.
Is ceroid lipofuscinosis neuronal, okay? Yeah. CLN. That's how the diseases are named. Yeah. CLN, and then there's a number, okay? So in the case, the one that this CSP is CLN4, And Carole?
Yeah, we are working on CLN3 and CLN12. So this numbering is more probably order of discovery.
But one important thing to emphasize is that these are rare diseases. Mm-hmm. And it's also important to convey to the audience that research in rare diseases is important. And it's important not only because we're going, of course, our research attempt is willing to help the patients with rare diseases. That is critical. But in addition, we need to know that when we are going into mechanisms, we're going also to open perspective for other non-rare diseases. Mm-hmm. Okay, this is important. We're here talking about neurodegeneration in general. And I can tell you that later on, you see that some of these protein of the genes are also involved in other diseases, such as, I can tell you this DNA JC5 has been also connected to Parkinson's disease. So because it has been connected with one protein, it's called alpha-synuclein, and alpha-synuclein is a major protein in Parkinson's disease. So when we go deep and we understand what this protein, these genes are doing, we are not only helping to open new avenues for the therapeutic for the patient with rare diseases, we are also opening new avenues to understand and to treat other more general diseases.
So to summarize what you said basically is that we should not think that if you're researching a rare disease, it's just only useful for that rare disease. And also that one gene is only responsible for this disease, because there might be actually way more involved. And in the research, you might discover also relationships that are useful for diseases like Parkinson's in this case.
Yeah,
exactly.
And there are very concrete examples of such genes, actually among the Batten disease genes, so the CLN12. For every gene, you have a copy from your mother and a copy from your father. And the rare diseases, so the more acute forms, they develop often when you have actually both the copy of your father and the copy of your mother that are mutated, either the same mutation or even a different mutation. But it means that really almost the whole function has been eradicated. But in the case of the CLN12, indeed, if you have both copies mutated, that leads to this form of Batten disease, or we call it can express a little bit differently, but many of these forms of the CLN12 disease are called the Kuforakep syndrome. It's a form of early Parkinsonism, let's say. And when only one of the copies of this gene is mutated, then it gives you a much higher risk to develop Parkinson's disease in late age. So it shows that there are totally common mechanisms that are at play, and in a way, in that case, it's really an acute form that you have when both copies are mutated. And if one copy is mutated, well, you maintain a certain function of that gene, but the fact that you have a little bit impaired function, that means that over a long time, you may then develop a disease later on.
You said a lot about mutation, and I just started thinking as well, because you said it might be the same mutation or a different mutation. How many mutations of a gene can you actually have?
You can have as many as you have codons in there. So each gene is composed of nucleic acid bases. So that's like the code, and every 3 bases, they code for one amino acid in the protein that it's going to form. And so each of these codons can have mutations. And you can even have mutations, there are non-coding parts in those genes. So there can be mutations in the non-coding parts. So that leads then to that you form more or less of the protein that is encoded by these genes. So you can have many, many different mutations in one gene.
So it basically means that you can have just one gene that you research all your life, and you still don't get far.
Look, this is an important question, because time's limit, and then we are, let's say, not only one gene. We're looking at a cascade, and now with Carole, we're looking actually sharing the mechanisms of all these diseases. But it's true, when you have a key question that is related to one gene, it's worth it to go deeper. I'm going to tell you one concept. When we're talking about mutation, it can happen 2 things. One thing that can happen when we want to understand what is the mutation doing. And then in terms of mechanisms, it could happen that the mutation is basically inactivating the gene. So you have the disappearing of the gene. Or one other thing that could happen is that the mutation is conveying a gain of function. We say a toxic function now to the new protein, So when you have a mutation, you still have the protein, but now it's a different protein that is toxic. Okay. So this is actually one of the advancement that we have done investigating this CLN4, this curve disease, because this is one thing that we have discovered, that is not the absence of the protein what is causing the disease, is the gain of a toxic function of a new protein produced by the mutation. So this is important.
And when is the moment when you say, okay, let's move to another group of genes?
I mean, when you really are satisfied with, the answers that you got in terms of, if you start working on a gene based on its sequence already, you can do certain predictions. And then you start looking in first on the protein level, then on the cellular level, then on the organism level. Does it stick with my predictions? And of course, you have to adjust your hypothesis, and because, not those things are failing that you would have predicted, etc. And you advance. But then if things start to just really fit together, so you have an understanding of the molecular function, meaning the biochemistry, the reaction, for example, that an enzyme with certain protein is catalyzing. And then you see that when you take away this protein or this gene from a cell, it's exactly like the compounds that that this protein should be working on that are not anymore there or accumulating. And then you have an idea, okay, if that is accumulating, then I think it's going to affect the cellular function in that way. And so if really you start seeing everything coming together at all the levels, then and also if it affects a disease, and by complementing then the gene function, the way you think it affects the function, by complementing it by drugs or other things, and you treat the disease, I think in that case you can start moving on.
Exactly. I completely agree. This long way. I mean, the gene is one part of the story. You need the gene, you need the mechanism, and you need, of course, to try to develop new therapeutic strategy based on this basic knowledge to help the patients. I would say that's really the way that we all would like actually to go through.
And I also know that, if you have your research group, it sometimes is also about the PhD students you get or the postdocs, and they have some certain knowledge as well that they bring with them, right? And it makes you go some other path too, right? So the inspiration of someone new that comes. And in this particular case, for your project, you also share some person, right, that unfortunately couldn't be with us today. But I think we should mention, right? So who is that and how is the sharing going?
So he's Santiago Lopez Vejinis. He's a doctor in biomedicine and has been a key person in this study and telling you about discovering the role of CSP mutation causing this calf disease. When we start to talk with Carole, we found that Santi could have this complementary approach, really a great opportunity. So he moved to Carole's lab, where he has been getting familiar with completely new things for him, such as zebrafish or metabolomics. And here, he was trained in electrophysiology, electron microscopy, image analysis, proteomics, and this is the expertise that then he's using you to collaborate in the zebrafish model with Carole. Carole, I don't know if you want to add. Yeah.
I would love to emphasize also that this it's really nice if you have common questions but complementary expertise, because you are really trying to join these forces and put together these complementarities to answer your question. And when Santi came to our lab, we have these zebrafish and we are looking at them mostly at the molecular level. So we basically just grind them up, and I mean the larvae, and it's ethically all covered, but we extract the small molecules and then look at those to understand more the metabolic questions. But Santi came, with this more looking at really the brain images, making sections, making very good microscopy on this. And so it's at a completely different level that he's looking, right? And of course, he was interested in our techniques, but he also taught us like how to handle our zebrafish to look at completely different aspects of our models. And, scientists, they always like to exchange, learn from each other, and with such complementarity and common questions, it's just an excellent opportunity.
Well, of course, you have to speak the same language as well, which is not always easy, right? But still, if it gets to very interdisciplinary research, then it is tricky, I know, because exactly if you talk to a psychologist about chaperone, I think that would be a different story, right? But I just realized, with this shared expertise, we did define metabolomics, we did define proteomics, but you also talked about electrophysiology. And I have to say that I don't know what electrophysiology is. So I'm missing one more definition, Rafael.
Let me tell you. So physiology is function. So we want to understand the function of the neurons. Neurons are electrical cells. So we need electrical recordings to understand or to measure, to study the physiology, the function of the neurons and how the neurons communicate with each other. That's the reason we call it electrophysiology. And we can then go and we can take single cells and we can take part of the brain and go to one single neuron and then look at the neighbor and look how two single neurons are communicating with each other, measuring very, very tiny electrical signals, because the tiny signals that are happening in your brain. And we can use that with a specific amplifiers, in a special device, we can measure this. And this is very accurate measurements.
So are these devices similar as the ones that you use just to measure electrical current, or it's special for this case?
Indeed, what we do is we measure electrical currents. This is a technique called patch clamp. Neer and Sagma got the Nobel Prize in 1991 because of these techniques. And you use very, very nice microelectrodes of the tip is 1 micron. And then you can then go directly with micro manipulators. That is, the sensitivity is nanometers. You go directly and then you see under the microscope, go to the neuron, and then you can measure how the neuron is triggering impulse and how the neuron is releasing and receiving neurotransmitters. Okay, so you can have a really accurate assessment of how the neuron communicate with each other.
It is fascinating, especially if we're talking about measuring that in mice models or whatever. It's just minute. Everything is so small, right? How big is a zebrafish, actually? I didn't prepare too much. I didn't check the sizes.
I don't know exactly. I think the larvae, they are at the order of a centimeter or something like this. So. Okay. But it's small fish, but you see them with the eye.
Okay, at least that, because yes, I have to visit your platform once to see. I don't know if that's possible, but it would be really lovely. I love, tools and equipment, and just because it's so difficult to talk about it if we don't really see it. I spend a lot of time, talking about exactly the exact measurements that people are taking. And if you don't see the sizes, your imagination is not really helping you for sure. Okay, well, I think this is the moment to solve the pop quiz question, because we are slowly wrapping up. So we're coming back to the numbers, as we said. So, Carole, can you remind us what the question was about? And then let's start with your answer.
Yes, so the question was, do you know how many genes we have in the human genome? And the answer is that we have roughly 20,000 different genes in this genome. So each of this genome copy, there's a copy of this genome in each of our cells. And as I said before, each gene is encoding for a particular protein that has a particular function in our organism. So it's 20,000, and those 20,000 proteins are orchestrating the whole functioning of our body. Since we know about this, we have now very elaborate databases that regroup all the knowledge that we have about these different proteins that we have accumulated also from before we knew the genome sequence. And what came out from that is that there is an impressive number of these proteins and hence of these genes for which we are not sure yet, or we even have still no clue about the function. And this amounts to, in the human genome, for example, to more than 6,000 of these 20,000 proteins. So it is roughly a third of our genome. And this is only the coding part of the genome, because there is a whole non-coding part which has maybe regulatory functions, etc. But I'm not even touching on this. But the coding part of the genome, which are represented by these genes, these are 20,000 proteins. And of these 20,000 proteins, there are a bit more than 6,000 proteins where the function remains completely unknown or uncertain, let's say. So still a lot of fundamental research to do.
A lot, exactly.
A lot of work ahead.
Okay, and going back, as I said, that was the first question. Now we have Rafael's question, and of course the answer as well.
So my question was, what is the number of neurons that we have in a human brain, and then which is the number of synapses that we have in a human brain. So neurons in one human brain is estimated to be 86 billion neurons, but let's say roughly, we can think about 100 billion neurons. But in order to have an idea the size of this number, we can compare that with the population of the Earth. And this means that a single human brain contains roughly 10 times more neurons than there are human beings on the entire planet. So this is a huge amount. So you can imagine, all these inhabitants talking to each other. You can imagine these neurons talking to each other. And you can think that it is roughly estimated that these neurons can form between 1,000 and 10,000 synaptic contacts of communication, though this means that the number of synapses is extremely high. It could be between 100 trillion and 1 quadrillion of synapses. So this is a huge number, and thanks to that, our brain has really these enormous capabilities.
Yeah, it is a fascinating number. Also very interesting, the comparison you gave us. Thank you for that, for the number of people in the world. I was thinking, Carole, when you were talking about the number of genes, whether we could compare it to a number of people living in Luxembourg, but it doesn't compare very well.
Well, we are small, but still a little bit more.
Yeah, it's a bit different, but we're not that many here, I would say. I mean, in the sense that there are less genes than we are in Luxembourg, but still we are not, we're not talking billions. That's what I meant, right?
Maybe Luxembourg people that really also speak Luxembourgish.
Well, you would be surprised, It's more and more, for sure.
Okay,
for
good.
sure.
That's good. Yeah, I noticed that also in my lab.
Yeah,
people coming in and starting to learn this small language.
Rafael, can you speak Luxembourgish as well?
No, not yet.
That's the right answer.
Yeah,
yes.
but I want to say that this is who knows, maybe in the future. Okay, so we're having a really very fruitful collaboration with Carole's lab, and I must say that I'm so grateful also to this initiative from the LCSB that is allowing this fruitful collaboration between LCSB and IBIS, and of course this idea coming from Professor Michel Henneka. It has been great. Okay, and we're so really happy to support this initiative.
Great, thank you for that. Yes, it's some of the collaborations here are really precious and really good, and I'm happy to be able to discuss with also international guests through that. Thanks to that. So, of course, I don't have a feeling we really discussed much, although we were talking for almost an hour. But at least a little bit, I hope that the listeners understood what you're working on and how much there is still to be done. Right? No pressure. Thank you very much, Carole and Rafael, for coming and sharing this part of your world with us today.
Thank you. It was my pleasure.
Thank you, Anna. Great pleasure.
And this is it for today. Don't forget to subscribe, to follow us, to check what we're saying. Of course, you can always suggest guests. If you have someone in Luxembourg or outside of Luxembourg that you think that I should interview, let me know. This was Silex, and my name is Hanna Siemaszko.